We are finally to the last portion of Anatomy and Physiology. Yes, we are going to be using the book Anatomy and Physiology by Wiley, or at least some of the objects and items that you're going to see in this presentation are going to come from the Wiley book. I do actually reorganize it a little bit to make it a little bit more sense, as well as a little bit of my background. I have a background in the reproductive signalings of implantation, all that stuff, so I kind of have an idea of what I want to go with. And I'm straying a little bit from what the book wants. I'm going to start off with fertilization to invading the uterus, which is a nice little episode of or a bit of time. Then we're going to go a little bit different. I'm going to go ahead and talk about the development of the baby, followed by the development of the support system. So kind of going forward in time and then reversing time and just focusing solely on the support system. With that, let's talk about this portion, about this video. This will be the first of 3 portions. We're going to be talking about fertilization. Fertilization, well, you kind of think of it as like the soil and all that stuff, but not so much. It's more along the lines of there's the mama egg and the papa sperm, which we talked about last week. But now we're going to figure out why on earth that needs to happen, why these two come together. So the sperm is actually going to swim up the vagina into the cervix. Now, bear in mind, remember that the sperm don't really have much energy to go off of, and it's kind of weak sauce as it goes because, well, by itself, it just can't. So it needs some help from the uterine walls to move along. The uterine walls contracting a little bit to help move it along into the uterine tube, where usually, most of the time, fertilization is going to occur. And that fertilization creates a zygote. Okay, so this is how it's looking. There's a couple of barriers going on. The main barrier that you got, if you remember, is this thing called a zona pellucida. This zona pellucida contains tons of little proteins, and because of it, even though a sperm might make it to the egg first, doesn't guarantee it a spot. In fact, it could actually drill a hole, only get so far in, and then only allow for the next sperm to get in. A little bit of Fall Guys going on, if you know what I mean. Now, to get a little bit more detail about this zona pellucida, just on the outside, there's a lot of different little proteins, zona pellucida 1, 2, and 3. In particular, there's a protein that we care about called the glycoprotein ZP3, which is a receptor, or aka the sperm goes, oh, I know this, and because I know this, I am going to throw up everything in my acrosome. And it throws up that stuff in that enzymes, releases it onto the zona pellucida, which eats up those proteins, so that sperm can hopefully maybe drill away into that egg as well and get to that membrane and deposit its nucleus in so that it can keep on going. If it is successful, the zona pellucida is going to do something more than just be a barrier. It's now going to become a good barrier, not just a bad barrier, a good barrier. It's kind of like social distancing back in the year 2019 when everyone was starting to freak out about the coronavirus. But the zona pellucida doing a really good job. This particular person, this is a gentleman, I believe he was from Germany, and he created a 6-foot radius apart. So if we're going to relate it to the embryonic development, this gentleman here is the fertilized egg, while these young ladies and other individuals out and beyond his little zona pellucida are the sperm. It's important to make certain that other sperm don't have a chance to get to it before it's prepared to say, hey, I can't receive, or pull away those receptors saying, okay, come on in. They got to get rid of those receptors. If those receptors are still there when a second sperm comes along, well, that egg's no longer going to be viable. So there's that zona pellucida. The outer edge of that zona pellucida expands away from that fertilized egg and creates this space between the egg and the zona pellucida, thankfully to make certain that polyspermiation doesn't occur. Because the egg has been fertilized, we have 2 pronuclei, one from the sperm and one from the female, both of which are haploid, meaning that they have just one copy of each chromosome. In order for it to become a viable individual, a zygote or something like that, those two need to merge together and become a diploid zygote. Diploid meaning it has the 2 copies, and away we go. And we're going to see some division going on. During day 1 after after division, we got some polar bodies and some blastomeres. Blastomeres are just a really fancy way of saying well, I'm going to call it an embryo cell. The polar bodies, we're going to remember that from the primary follicle cell, it went and paused after the first round of meiosis, and it's going to continue only after fertilization. Once fertilization's occurred, that polar body sheds off, we start dividing, those polar bodies are just going to go away. Day 2, we're going to start seeing those cells divide. An important note to know about these, if you yoink one of these cells away and like let it float by itself, it could become an identical twin. So this is a maternal twin. If it were a paternal twin, you'd have 2 eggs getting fertilized by 2 different distinct sperm. So they're not genetic. The paternal would be non-genetically identifiable, while the maternal would be exact duplicates. This is the one that they look exactly the same. At day 4, you get something called a morula. This is when it's like 32 plus cells. You can't really count the cells very easily so much anymore. It's just a solid mass of cells. It's just clumped. There's not really any seemingly any organization to it. Finally, it gets its act together, this clump of cells, and says, you know what? I want to be a ball of some sort. I like balls. Well, it became something called a blastocyst, because we're not going to call it a ball. Blastocyst, there's a nice hollow space in the middle, except for this like little chunk of cells right here, and then there's something out here that's acting like holding the liquid inside that little blastocyst. All this is occurring within the uterine tube. Usually by the time it gets to that uterus, it's actually a blastocyst. So the question is at this point, which part of that little blastocyst is actually going to be baby? Which part of it's going to be supporting baby? And this is something you should be asking throughout the development of this child for the whole 8-9 months it's going to be there. For us, starting with the blastocyst, this thing right there is called baby. This is the inner cell mass. The rest of it is going to be called the trophoblast, which is eventually going to become interacting with mama's cells, eating away at mama's cells, moving away, reorganizing it, and hopefully setting up a support system that it's going to need. We do need to name these cells in their appropriate names. These things right here that's actually become the baby, the embryo, aka the inner cell mass, is called the embryoblast. Hey, how convenient. Embryoblast. Wonderful. The rest of it, this outer cell mass, is going to be called the trophoblast. This is what's eventually going to become the chorion. This is eventually what's going to become the placenta and all that stuff. It's the thing that's going to keep baby alive. It's going to keep the embryo and fetus alive. Now, here's the problem. We're in a blastocyst, but we're still in that zona pellucida, that nutrient-dense, protein-dense shell. We have to escape from it. But we're going to there's a lot of complex situations, signalings, and metabolites of estrogens, progesterones, and prolactins saying, hey, this should happen at this time, but we're not going to talk about that. All we know is that when it is ready, it is going to leave that zona pellucida via some signaling, and then voila, c'est magnifique. These cells on the outside are now going to be able to interact with the mom's uterus, the stratum functionalis, if you remember. And once it touches down, it's going to do something called implantation. Now, remember thinking of implants. If somebody gets an implant, let's say a breast implant, yes, they shove something into that woman's chest cavity, and then they put it in. That's exactly, I know that's kind of graphic, but that's kind of what the blastocyst does. It says, oh, this is a nice place, and it like digs in and then like buries itself. This is happening about day 6. At this point, this is where we want to discuss the different parts of the embryo. I'm going to pause here. I'm going to let this be what we just talked about, be its very own video, because that was a little bit of an information-dense slide, and we're going to come back to this in the next part. This will be part 2.